Gram staining was invented in 1884 by Danish scientist Hans Christian Gram and is still a primary method for distinguishing between bacteria even today. Welcome! In this edition of Keio Basic Bytes, we will perform a Gram stain to empirically demonstrate the ins and outs of the protocol, offering as well some of the theory behind this technique. We begin by labeling each slide at the correct end in pencil. Ink labels will not survive the staining process, and remember, always label the same side of the slide as the bacteria. Carefully place each of the labelled slides in the cradle provided. It helps later if the slides are all facing the same way and are spaced as far apart as is practical. The first step is to immerse the entire cradle in Gram's Crystal Violet for a period of 60 seconds, as shown by the timer in the video. The crystal violet binds to the cross-linking in the peptidoglycan cell walls of the bacteria. After the minute, carefully remove the cradle, take it to the sink area and proceed to rinse the assembly under the tap for a few seconds, watching for splashes at all times. Then, tap the whole assembly onto laboratory tissue paper to drain. At this point, all the bacteria will be a uniform violet colour, regardless of whether they are gram-positive or gram-negative. Moving on to step 2. Using the droppers available in the lab, carefully flood the faces of the slides with Gram's iodine, whilst holding the cradle over the sink area, always with a mind to safety. Then, fully immerse the cradle assembly in Gram's iodine for a period of 60 seconds. The addition of iodine promotes dye retention of the bacteria by forming a chemical complex with the crystal violet from the previous stage. Once the time is up, remove the cradle as before, take it to the sink area and rinse the assembly under the tap for a few seconds. Again, tap the whole assembly onto laboratory tissue paper to drain. The next step is a decolorization step. This is critical and must be timed correctly, otherwise the crystal violet stain will be removed from all bacteria. The aim of this step is to allow the acetone decolorizer to remove unbound dye and separate gram-positive from gram-negative bacteria. With this in mind, immerse the cradle in acetone for a maximum of 30 seconds. Even a few seconds longer will be too much. Once the time is up, remove the cradle as before, take it to the sink area and proceed to rinse the assembly under the tap for a few seconds. For a third time, cautiously tap the whole assembly onto laboratory tissue to drain. At this point, the gram-positive bacteria will be a violet colour, but the gram-negative cells will be colourless. This can make them difficult to visualize, as you will see later. In the final step, we will immerse the cradle in a counterstain, safranin, for 60 seconds to stain the colourless gram-negative bacteria pink, enabling differentiation between these and gram-positive bacteria. Once the time is up, remove the cradle, take it to the sink area and proceed to rinse the assembly under the tap for a few seconds. After the final time, tap the whole assembly onto lab tissue paper to drain. After the final tap, you should carefully remove each slide from the cradle one at a time and wipe the back of it to speed up the drying time. Caution, do not wipe the front of the slide or you will wipe off the cells you just stained. After wiping, allow your slides to air dry for a short time and view under oil immersion. If necessary, see Keio Basic Bytes oil immersion video. This is not a shameless plug. Some bacteria, such as the mycobacteria, yield a gram-variable pattern after staining. Here, mixes of both pink and purple cells are seen, and this can be hard to interpret. Likewise, some cell walls are sensitive to breakage during cell division, whilst others have decreased peptidoglycan thickness during growth. This can sometimes result in false positive or false negative results being observed when older cultures are used. Come with us now, down the microscope, as we examine some of the things you will see. We will look at examples of both unstained and stained bacteria. Here you can see the unstained slide of Bacillus subtilis. You can see that when stained, the bacteria are much easier to identify. Bacillus subtilis are gram-positive rods. This is an unstained slide of Escherichia coli. These are much harder to see than bacilli, as they have no capsule. Once stained, you can see the bacteria become much clearer. These are light pink bacteria and are gram-negative rods. Here we have unstained Staphylococcus aureus. Note they are virtually impossible to identify at this stage. Once stained, these bacteria are now readily identifiable as gram-positive spheres. We hope you have enjoyed another Keough Basic Bite.